everybody. I'm Marco, and I will present a project that I'm working on it with my company Sequentia. But let's break the ice in the beginning. What about Sequentia? Sequentia is a bioinformatics company that works on many different omics data. We have really advanced expertise in healthcare, agriculture, and wide biotech industry. We are based in Barcelona, and we really enjoy it a lot. We have the beach, we have the vermouth, we can do a lot of stuff there. We have customers in more than 65 countries, and we delivered more than 500 bioinformatician projects. Moreover, we have more than 1,500 cloud software users. And this is our team. And as I said to you, we enjoy a lot of beach. And with this kind of group, we can organize also a lot of beach volley play. But yeah, I'm there in the middle. I'm not that athletic, so I just passed the beer and I am the judge of the tournament. But we also work a lot. We don't just drink vermouth and we just don't drink and we play with the volleyball. We publish more than 70 papers in top scientific journal. More than 30 are involved in green tech papers, and more than 22 are involved in the human health and all different analysis with omic data. If you want some more information about the things that we have published, you can use this link. So what do? We perform many different bioinformatics analysis, but also offer to the clients and to the user many different solutions, starting from the custom softwares, the cloud software, the consulting, and also training. If you are interested in do some training with us, I have some more Depliant and a contact card, so you can write us and maybe, if you want, participate in our life. Our technology is innovative on multiple levels. We go through the platform to the proprietary infrastructure, to the bioinformatics, and also to the products that we give to the user to perform their analysis and to visualize their own data. And as I said, we work with many different omics data, going through the genomic data, transcriptomic, metagenomic, and epigenomics. So we have a really good metadata integration and omics integration. Our division is really wide. We go through the healthcare, as I said before, to the industrial part, but also to the agriculture field. And this will be the topic on which we will have this presentation today, since with our application, we give to the scientists the opportunity to study better the crop protection nutrition, to improve all the analysis that might be involved in the plant breeding, and also the agricultural microbiome. We have several partners in all the world, going through the cows, to the CRAG, the CA, and many different companies, and we work with them. And we have won many different awards from European, from national, and local organizations. So after this brief introduction, let's go deeper in the project that we are working on. And this is GRAPEDIA. GRAPEDIA basically is a consortium of different researchers inside the world, going through the University of Davis to the MAC in Trento and the University of Padua, and of course, also Sequentia and many different universities in Spain. And this is basically a portal that will be useful for all the Grapevine researchers. Why Grapevine? Mainly because it's one of the most important crop in agriculture, basically, and also for the economic information that could give us for the production of wine and production of fruit. But it's really important, this concept, because we don't have right now a portal that might be useful for the researcher. And also because Grapevine is really sensitive to many different abiotic stress. So we need a portal that could help us to improve the production of new varieties. So just a little bit introduction of Grapevine. It's Vitis vinifera, this Latin name. It's worldwide important for economic reason and also for drinking reason. There are more than 10,000 known varieties, but let's focus on this number. Only the 0.3% of all these varieties are really important in the half of the world's vineyards. As we can see here, the production is really strong. We talk about 26.25 million metric tons only in the 2020-2022, and the economic net, it's really strong. We're talking about $9.31 billion just in 2022. So this is a chart from 2019 where we have all the main important countries producing grape. And we have China, Italy, United States, Spain, and France that are the most important. But the community, of course, needs a portal for a species that is a non-model. So what do we want? A portal like TER. TER is basically the database in which all the information about Arabidopsis italiana are stored. When do we want? Yesterday. They really want it. And so with GRAPEDIA, we're trying to give them a portal like TER that might be nicer than TER. So basically, GRAPEDIA is the innovative portal that integrates all the biological knowledge about grapevine, but also all the genetic, genomic resource, and also customized service that with Sequentia we want to give to the user and to the scientist. This is basically the ambitious project that we have. This is an example of a dashboard that we want to give to the community, and will be basically gene-centered in the beginning. We are going through the exploration of catalog of gene cards. We can give to the user co-expression networks, heat map, gene expression atlases, and genome browsers. But this is just a little example of what we can do. Basically, the scheme is cyclic. We are going through the community. We have researchers that can give us their own data. We have breeders that can give us information about the phenotypic traits and all the information that can be used for the production of the portal. And wineries, of course, because as I said, wine is one of the most important elements for the production of this portal and also to the improvement of the crops of the varieties. Through the community, we have the data, exhaustive data that can go through the gene catalog, genomes and transcriptome, QTLs and SNPs, phenotypes, and in the future, we will also integrate information about metabolites, epigenomic, and metagenomic. This data will be stored in the federative database system, and through APIs that we will develop and we will create, we will connect the database to the interface to the portal, giving to the user open access feature, but also tailored services. And through this, we go back to the community. So this cyclic element can start from the community and go back to the community. This will be the scheme that we thought about to produce this ambitious project. We go through the community. So basically, in this point of view, as Sequentia, we are not producing data. We are collecting data from the community. All people and all group of research that want to give their own data can contact us, and we will inside them in the federative database. We will store all of them, transform in a format that can be useful for the service, and then store in the database of GRAPEDIA. And through REST APIs, we will use this data to produce all the service going through the web, the dashboard, and the workflows. Let's talk a bit about the resources. Right now, we are at prototype level. We don't have all the genome. We don't have all the assemblies for all the varieties that are known for grapevine, of course. And we will focus our work on the reference genome that is PN 40024 version 4. That for matter of simplicity, for right now, I will call like PN. So PN basically is a resequencing of a Sanger assembly completed with biodata, and the annotation is de novo and also include reciprocal matching with the version 3 of the PN. We have also catalogs where do we have all the functional annotation on the version 3, and also validation level scheme where do we have all the information about the function of the genes and also how much is related to a phenotype that we want. This is version 3, but we will have soon with training school that we're organizing with the GRAPEDIA consortium a version 4. And we have all the expression data that are collected from the public database and stored in a Compass interface called Vespucci that is made by FNAC. Talking about the genome assembly and annotation, it is basically just a chronological time flow. We started everything with the Sanger assembly in 2007, and then with all of these steps going through the improved scaffold mappings and the resequencing, we arrived to the version 4 that is resequenced and corrected with PacBio data. And this was performed by the group of Welt that has also members that are vice chairs of the GRAPEDIA consortium. And for this, we also performed the annotation, and we have the remapped annotation through liftoff overlaps, and all of these were produced with a prequel consortium that was called Integrape, that is basically the main one, and GRAPEDIA is the sequel of this consortium. Talking about the gene catalog, this is more it is a crucial object, a crucial and important point of view, because we start from the published data and we have done a really deep literature search in order to annotate all these genes in a specific way. So when we encountered new gene family, we validated them through the annotation and also with public RNA-seq data. And we produced the GRAPE gene reference catalog that can be exported. And we have right now a Shiny app called in BitViz platform that can be used by the user to identify and evaluate the gene for the version 3 of PN. And this is the Compass graphic QL of Vespucci, where all the information about the expression level are stored for the version 3 of PN. And with this information, the user can download all the data and perform their own analysis. Talking about the connection to the database, we want to have a connector that might be really useful and really simple to use. We are working with this software called Airbyte that through Python scripts can connect and extract all the data by the web and transform them in a format that the database can be called and can store all the information and load it on the PostgreSQL database. And we are not talking just about assemblies or annotation file like GFF. We are also talking about URL, links, spreadsheet that can be stored and used for the creation of the portal. This is the basic concept about the federative database that we want. We have the source data and the database that can be connected to GRAPEDIA through the ATL algorithm, and then have the current data stored in data federation, or through ATL we can store them in data warehouse and data marts, and have also the historical data. And then through this virtual database that we have, connect them to the portal and give the possibility to visualize all of them. With Sequentia, we are working on the services that we can give to the users and also in the management of the database. In particular, we will work on the API development. We will integrate all the open source tools that can be useful for the community. We will develop cloud softwares, customized algorithm, and also ensure the security and the maintenance of the portal. Here we have just a few of the software that we have developed with Sequentia. We have AIR, that is basically a pipeline that can perform RNA-seq and differential expression analysis. We have Genomics that is involved in the RNA analysis for agriculture. and Gaia, that is our metagenomic data analysis software. So here we have all the service that we will give to the community. We will produce the website, advanced, responsive, and accessible, the dashboard that can give the users the possibility to integrate data and visualize them, and can be free, custom, and open source. We are working with the introduction of the GBrowse version 2. We will also add tools like Drago, that is our database of plant-resistant genes, and open source tools like BLAST. Moreover, we will give to the user the possibility to upload, download, share data, and also manage the profile and the data that will be stored inside the portal. And moreover, this is one of the most ambitious parts that we are working on it, and we are also organizing a training school that will take place in March. We will also produce pipelines to run bioinformatics analysis using Nextflow and Docker. So we already saw this diapo, this slide, and what does it change here? Which change the product. Here we have the GRAPEDIA services, and here we will have all the information that I said to you before. We will go to the infrastructure, the informatic algorithms, to the product, the final product that will be the GRAPEDIA service that we will give to the community. Here we have an example of the GBrowse that we will integrate to visualize the genomic data, the BLAST that can be used by the user to perform all the analysis. We have just an example here, but at the final level of the GRAPEDIA, we will have more than 20 varieties and more than 20 assembly that can be used by the users to perform their analysis. And we will also integrate software that we developed, like the database with Drago, that is the algorithm that use our database of plant-resistant genes, and integrate with Vitis vinifera data. And here we have just a little example of how Drago works. We start from the plant transcriptome of proteome through a series of algorithms like coils, TMHMM, and custom hidden Markov model. We go through the identification of the CC potential, the transmembrane signal, and the resistant domain evidence in order to have putative resistant genes identified by our algorithm. And basically this will be the output. We will have an ID from the database, the gene name of the gene associated to this ID, and all of information about the class and the visualization of this gene. This one, instead, is the green non-coding database. We worked on it a few years ago, and this is basically a database for long non-coding RNAs that we developed in Sequentia. And this is the page for Vitis vinifera taken from Phytosome 13. So if I don't remember bad, this is on the version 3 of the PN. And here we have all the gene aliases that are connected to the database, the position, and the number of long non-coding RNAs that might be present here. And how did we develop this algorithm? We started everything from the transcript, and we filtered all the transcript that had a length higher than 200 nucleotides and showing ORF lower than 120 amino acid. And then through blastics and the calculation of coding potential, we were able to identify putative long non-coding RNAs. Through this, we go to the part B of the algorithm, where we perform a series of analyses like BLAST and microRNA validation and CM scan in order to identify all the possible RNAs that are not long non-coding, filter them out, and then have the final set of long non-coding RNAs. So basically, this is the origin of GRAPEDIA. As I said to you before, we started everything through Integrate, that was basically the cost action that give a series of opportunity like the production of tools in the resource hub, the production of reference genome, annotation, and so on. the production of the gene catalog that we have seen before, the guidelines and the cookbooks for the identification, for the analysis, omics, and phenotyping, and also the possibility to produce new resources through other community, like the transcriptome explorers that we showed, like Vespucci, and also the genome catalog called Genomics. And the idea with GRAPEDIA was to connect everything that was produced by Intergrape going through the community that can use the resources, that can use the assembly, add new resources with the reference catalog with guidelines that are known by the community. And so this is the idea of the logo that the consortium had. So to build GRAPEDIA, we need a really strong community, and with the cost grant that allowed the presence of GRAPEDIA inside the Grapevine community, we are offering working group meetings, training school, short-term missions, dissemination activities, and different business plan. This is a time chart of the grant period that we have. Right now we are here. We are working on the prototype of GRAPEDIA. And in this period, we are working on modules related to the genes, to the genomes, and the QTLs, and we are producing the basis of the portal. In the future, we will also add multi-omics information, the information about the cultivar, the germoplants, and so on. And lately, we will introduce more information about the modeling for climate resilience. So this is the team that is quite huge. And I don't know why they did not put me here, but it's okay. I'm not so picky. And the sponsor of this work are the Cost Action, Sequentia, of course, Novatech, and Gallo Wineries. Here, if you want, you can have more information about GRAPEDIA. We have the website, we have Twitter, we have WeChat, we have Bilibid, that I don't know what is it. And I will try to talk with the chair and add also Snapchat and Instagram and TikTok. I don't know if it would be nice, but I don't know. So the future in the Grapevine relies on the community, and I will really thank you to give me the opportunity to present this work. Thank you.